Bismillahirrahmanirrahim. Today the topic of my discussion is cholesterol synthesis and its regulation. What is cholesterol? Cholesterol is a derived lipid with 27 carbon atoms in this compound and hydroxyl group at the third position, and there is a double bond between the carbon number 5th and 6th of the cholesterol molecule. These are the properties, some of the properties of the cholesterol molecules, that it is an animal sterol and it is a precursor for bile acid, and we know that where the bile acid is located, it is secreted from our liver, vitamin D and steroid hormones. So it is a precursor for all these. It is a compound of component of plasma membrane, and we know that the plasma membrane is composed of proteins, lipids and cholesterol molecules, and requires for the transport along the plasma membrane. So these are the important functions of cholesterol in our body, that it is an animal sterol and it is present in the main component of plasma membrane. That means it is present in every cell of the body and it is required for the transport of materials along the plasma membrane. It contains cyclopentanoperhydrophenanthrene ring, that is CPPP. This is the name for the cholesterol molecule, cyclopentanoperhydrophenanthrene. These are the names of the rings of the cholesterol molecules. Rings, there are total 4 rings in the cholesterol molecules. You can simply go on to the Google, and the Google for the structure of the cholesterol, you will have the structure of glucose there, structure of cholesterol there. You can see the structure of any compound there, like glucose, acetyl CoA, whatever you need, you will find it at Google. But for the structure of glucose, when you find it at Google, you will see the details, that where is the double bond and where is the methyl group, and you will see that there are 3 rings that are 6 membered, and the one ring is the 5 membered ring, that is the 5 carbon are present in that ring, and the 3 rings are 6 carbon containing rings. So rings A, B, C are cyclohexane, that means they contain 6 carbon atoms, and the 4th ring is the cyclopentane ring. The synthesis of cholesterol occurs in all the nucleated cells of the body, that means in the nucleated cells of the body, that means especially in the liver, adrenal cortex and intestine. And what is adrenal cortex? It is a part of kidney. So it is synthesized mainly in the liver, in our intestine and in the kidney. It is a cytosolic pathway, and some steps occur in the endoplasmic reticulum. The synthesis of cholesterol is a cytosolic, that means it occurs in the cytoplasm of the cells, and some of the steps occur in the endoplasmic reticulum. Especially the steps catalyzed by the HMG reductase, which is a rate limiting step in the cholesterol synthesis. And what is HMG reductase and what are the steps, we will see here. I will show a chart, a full diagram, that how the cholesterol is synthesizing and how the enzyme is acting on it. There are 5 steps in the synthesis of cholesterol. So first one is the HMG CoA hydroxymethyl glutarate CoA. So HMG is the hydroxymethyl glutarate. When HMG synthase, as we have seen here the name, the HMG is synthase, so that means the hydroxymethyl glutarate synthase. So HMG coenzyme, that means hydroxymethyl glutarate CoA, formation of mevalonate, conversion of mevalonate into the isopentenyl pyrophosphate, formation of squalene, and conversion of squalene into the cholesterol. So here is the pathway. Starting material is like acetyl CoA. It requires NADPH and ATP, and the one main thing is here that 60% NADPH that is required for the cholesterol synthesis comes from HMP shunt, and you can listen to the details of HMP shunt in my next lecture, yet how HMP shunt generates NADPH. This is the whole chart for the synthesis of cholesterol molecule. Here we should memorize that 2-acetyl CoA are utilized for the conversion, for its conversion into the glucose. It is a detailed step and it requires a lot of steps that how cholesterol is converted, is synthesized from acetyl CoA. So acetyl CoA is converted into the acetoacyl CoA, then acetyl CoA is converted into the beta HMG CoA, then beta HMG CoA is converted into the mevalonate, then mevalonate is converted into the 5-phosphomevalonate, then it is converted into the 5-pyrophosphomevalonate, then this is converted into the 3-phospho-5-pyrophosphomevalonate, then that is converted into the isopentyl pyrophosphate, then dimethylylide pp, and then geranyl pp, then ferricyl pp. You can learn this, and you can also form a mnemonic in your mind from the first letters or whatever you want, so that you can memorize the whole stuff. Then it is converted to the squalene, which is converted into the lanosterol, and lanosterol is then converted into the cholesterol. So I have also added the enzymes that are required here. The enzyme is thiolase HMG CoA synthase. Here the one more molecule of acetyl CoA is used, and here coenzyme A are also utilized. In the third step, HMG CoA reductase is utilized, and this is the main step of this whole process, that HMG coenzyme reductase is required. And I have also told you earlier that this is the rate limiting step here, so the whole process depends on this step. Then here NADPH is required, and from where does the NADPH came? NADPH came from HMP shunt, that you can listen in my next lecture. Then mevalonate is converted in the 5-phosphomevalonate by the help of ATP. So because by utilizing ATP, a molecule of phosphorus is added, and a kinase enzyme is required, so that we know that the kinases are required for the transfer of phosphate groups. Then 5-phosphomevalonate is converted to 5-pyrophosphomevalonate, and here also molecule of ATP and the kinase enzyme is used. Then again a molecule of ATP and kinase enzyme is used, and it is converted to 3-phospho-5-pyrophosphomevalonate, and then decarboxylation occurs and a molecule of carbon dioxide is released, and then isomerase enzyme is required. Then IPP 5 carbon, that means you know the name of this compound, that is the isopentanophosphate group. Then you know that this is a pentanophosphate, so this is a 5 carbon compound. So then it transfers its phosphate group, and the 5 carbon compound is converted into the 10 carbon compound. Then with the help of IPP, IPP again it is utilized, and then the 15 carbon compound is formed, because IPP is the 5 carbon compound, as we have seen here. Then this compound ferricyl pyrophosphate is converted to squalene, which requires NADPH. And from where does this NADPH came from? NADPH came from HMP shunt. So it utilized, this step also utilized ferricyl pyrophosphate synthase. Then it is converted to squalene, that is a 30 carbon compound. Then NADPH is required and oxygen is used. Epoxidase cyclase enzymes are used, and it is converted into the lanosterol, which is a 30 carbon compound. Then due to the removal of 2 carbon dioxide molecules here, and here also the molecule of NADPH is utilized, oxygen is utilized, and a molecule of formic acid HCOOH is released here. So it became, 3 carbons are removed, 2 from carbon dioxide and 1 from formic acid HCOH, so it became the 27 carbon compound. So that is the whole process for the synthesis of cholesterol molecule. So now we can see that how these steps are regulated, how this pathway is regulated. This pathway is regulated by 2 states, dephosphorylated and phosphorylated state. The enzyme HMG CoA reductase is active in dephosphorylated state, so insulin maintains the dephosphorylated state, and also thyroxine. So glucagon inhibits HMG CoA reductase. So there are 2 hormones required here, that is the second modification is feedback regulation. In feedback regulation, the cholesterol inhibits the HMG-CoA reductase by inhibiting the transcription process. So feedback regulation is at genetic level. Dietary cholesterol has a minimal role in the inhibition of cholesterol synthesis. Degradation of cholesterol. It is a precursor of three important compounds in its degradation, that is bile acid, steroid hormone, and vitamin D. So that was all about the cholesterol metabolism. If you have any query related to this topic, you can ask me in the comment section. Thank you.